In protein synthesis, there are two major steps, transcription and translation. Transcription is the process of changing DNA into RNA, which takes place in the nucleus. The transcription process is broken down into three steps: initiation, elongation, and termination. DNA is made up of four nucleotides, adenine, guanine, cytosine, and thymine, which pairs up with each other. Adenine with thymine and guanine with cytosine. These nucleotides make up genetic codes. During the first step, initiation, several transcription factors bind to the Tau box. The Tau box is a DNA sequence found in the promoter region. Once the DNA is bound to the Tau box, an enzyme called RNA polymerase unwinds the DNA double helix with the help of the transcription factors. This leads to the beginning step of elongation. Elongation occurs as RNA polymerase moves down the DNA strand from the F' end to the 3' end. It creates copies of the genetic material. The copy it leaves behind is messenger RNA. The mRNA strand is complementary to the template strand of DNA. It has opposite bases, guanine instead of cytosine, but rather than substituting thymine for adenine, uracil takes its place. When the transcription reaches the 3' end of the DNA strand, the mRNA detaches. The process is then terminated, and the result is the complete strand of mRNA that is ready for editing.
After the genes are transcribed, RNA modification occurs. A 5' cap is added on the end of the messenger RNA strand to allow it to leave the nucleus easily. The 5' cap also protects it from being damaged as it travels through the cytoplasm. On the opposite of the 3' end is a poly A tail, which is a long chain of adenine nucleotides meant to increase stability of the molecule and further protect the molecule. After this, introns and exons are part of the gene. Exons code for proteins while introns do not. Therefore, introns are removed with the help of SNRPs during a process called splicing. Finally, the messenger RNA is able to carefully and safely move out of the nucleus and into the cytoplasm.
Now translation begins. Translation takes place in the cytoplasm and also consists of initiation, elongation, and termination. During initiation, mRNA moves through the ribosome. It has a small subunit and a large subunit. Every 3 nitrogenous bases in mRNA called codons code for one specific amino acid. tRNA molecules are taken to bond with ribosomal subunits along the mRNA strand. Elongation begins with the first tRNA molecule pairing with a start codon AUG, or methionine, at the P site. An enzyme called aminoacyl tRNA synthetase uses ATP to attach amino acids to the end of tRNA anticodons. Then a new tRNA with anticodons enters the A site. An enzyme called peptidyltransferase creates peptide bonds between two amino acids attached to tRNA as amino acids from the A site attach to amino acids from the P site. GTP is used in this process. To form a protein, the amino acids are linked by dehydration synthesis to form peptide bonds. tRNA from the P site transfers over to the E site. Then, tRNA from the A site moves to the P site. The new tRNA strand comes in, and the process continues until a stop codon reaches the A site. Termination begins when the release factor detaches the polypeptide from the ribosome. Protein synthesis is now complete.